All right, welcome to another problem solving session. Today I'm going to talk about game theory and embryos. And you might ask, why does game theory apply to embryos? And the answer to that is that we can use game theoretic strategies, game theoretic mathematics, game theoretic formulations to understand different aspects of development in embryos. So I'm going to talk about one paper we did with embryogenesis and another paper we did with connectomes, emerging connectomes in development. So let's start with an embryo. So an embryo, of course, we start with a one cell embryo, and that immediately divides into two cells. And we can see that, like, immediately upon a two cells embryo, we have maybe basic polarity established. So we have an anterior and a posterior end. If we map that to a lineage tree, we get an anterior and a posterior end of the lineage tree. Or we can have some sort of division in the lineage tree where we have two sublineages that will go on to divide and fill in basically these spaces where this lineage gets established. Now, in what we're trying to understand is how these lineages establish themselves as sort of an interaction between the different sublineages. So if we go to the 8-cell stage here, this 8-cell stage, so we have an anterior and a posterior end, and we have two division events apiece. So we end up with this 8-cell lineage tree. Actually, 4 division events, but we might also consider that there are these strategies that emerge that, one cell, one part of the embryo might be favored over another in terms of the amount of space it takes up in the embryo, or one part of the embryo might divide faster than another part of the embryo, or we might get asymmetric divisions. And so in this case, we can use something called first mover strategies. And people have used first mover dynamics or strategies in biology, in a number of different ways they've applied it. It's basically a tool from economics. Sometimes it's a Stackelberg equilibrium. And this is something that people use in markets a lot to understand like advantages of first movers in a marketplace. So if you come out with a product first, if it's an innovation in the area, you have a first mover advantage, meaning that you are the first to be able to innovate, the first to be able to capture market share, the first to be able to sort of take what you want from the setting that you're in. Similarly, with something like tic-tac-toe, this is a game that has a distinct first mover advantage. If you've ever heard people say, if you play tic-tac-toe, you have to put your X or your O in the center square. So if I'm X and you're O, I put my X in the center square. You put your O here. I put my X here. You put your O here. I put my X here. You put your O here. I put my X here. We're constantly kind of in this equilibrium where, the first mover has some sort of an advantage, and then the second mover is constrained by the first mover, and so on and so forth. So that's kind of, one way to look at that. You have an embryo where you have, say, 2 cells. There's an initial division event. And let's suppose that this anterior end is much more So let's suppose that the anterior cell is much bigger than the posterior cell, something like that. So if we have an asymmetric division in terms of size for these two cells, this anterior cell has a sort of first mover advantage in that it's larger, and when it divides, if it divides in half, it'll retain that advantage in terms of how much of the embryo it's occupying. So spatially, it has a first mover advantage. We can also look at this in terms of lineage tree, where we have maybe like an temporally asymmetric cell division where we have, say, our anterior or posterior cell, and the anterior cell divides first, followed by the posterior cell. So this time point up here would be a first mover advantage in terms of a division. So if you look at the divisions in a lineage tree, you'll see that there's this advantage because it can simply divide and migrate. The cells can simply divide and migrate where they want to sooner than this one. That's what we mean by first mover advantage. So there's this paper we published way back in 2018. This is Origins of the Embryo, Self-Organization Through Cybernetic Regulation. This was published in Biosystems. The paper goes through a number of different models that we can use to understand the embryo and its self-organization. One of these models happens to be this tic-tac-toe game that I talked about earlier. So if you read the game theory literature, they alternate between noughts and crosses and tic-tac-toe. This is basically it, where you have this game where you have two players, the two players being two cells in a binary division. Sometimes it's the basic polarity of an early embryo. Sometimes it's two cells dividing. Sometimes it's two regions dividing. You can divide up things as you want them. But basically, there is some first mover advantage when there's an event that, either it could be cell division or it even could be cell differentiation. If that's something that is, something you can definitely model differentiation in that way. So this figure kind of walks through this basic idea where, it shows the tic-tac-toe game, and then it shows these different moves in sequence. So in this case, the anterior end here, number 1, is bigger than the posterior end, number 2. So the anterior end has this first mover advantage. There is also a division earlier than 2. So there's an additional first mover advantage from that. And it tends to occupy more of the embryo. It tends to occupy more space. It tends to occupy with more cells sooner. So you get 2 dividing, and then you get 1 dividing again with an asymmetric division in terms of like producing 1 cell instead of 2. And you can see that cells from the anterior end outcompete the cells from the posterior end, and that would have then consequences in the resulting phenotype. You could also do this with cell differentiation. If you think about cells differentiating into different cell types and subtypes, you can have one, say, have a first mover advantage over type 2, and maybe that's something that differentiates into many different subtypes, whereas number 2 differentiates into fewer subtypes, and those maybe one type of cell serves as the main type of cell in the organism, and then number 2 serves as the support type cell. So you can have this sort of trophic types of trophic organization in tissues, or cell types in an organism, in an embryo, and it can explain origins of different tissue types, different cell types in an embryo, and that can translate into the adult phenotype eventually. So this is this kind of, the way we're talking about game theory here is we're talking about in terms of origins. So if we have, these origins of, these early first mover events, we end up with this sort of advantage that carries over into the later phenotype, and it can explain asymmetries, but it can also explain sort of how things are kind of packed into a phenotype. It can also explain how the timing of things, how things emerge and when they emerge. And this is figure 5, which shows it's a theoretical graph, so it doesn't really map to any data, but it just shows the number of turns that are taken by the first mover and the number of possible moves that it constrains the second mover to. So the second mover, when we have that first sort of X in our tic-tac-toe, and then we have a number of options that we can take, but then as the game proceeds, you have fewer and fewer options, and this kind of constrains the behavior over time, this configurational constraint that's defined in the paper. So it definitely does constrain things for the second mover, but also for the first mover as well, because the first mover, there's an accumulation of first mover advantages. It's not just that lineage that has the advantage, but it's the first individual in that lineage, how it kind of sets up everything. So there's this role of sort of constraint that or developmental constraint that occurs as well. In the first paper I talked about, this was applied to embryos and cells and cell divisions and cell differentiations and origins of, say, like a phenotype. And so you can use the first mover model, the game theoretic model for that, but you can also use it for sort of the formation of things like connectomes. So it's not just about cells, it's about the connections between cells. It's about developmental structures that emerge and how those emerge over time and the dynamics that are shown over time. So remember, we're still talking about these first mover dynamics, this Stackelberg equilibrium. And so in this paper, this is raising the connectome, the emergence of neuronal activity and behavior. C. elegans, this is from 2020, talking about something called connectogenesis, which is how the connectome starts to emerge, how it connects from cells that are differentiating and coming into their adult positions in the organism. So we have these cells that sort of take their place. They're born, they're differentiated, and then they have to connect to one another. So when you have a connectome of different neurons, they have to find connections. There's this axonal guidance that sends an axon out to another cell, and then there's a synaptic formation so it can communicate with that other cell. And so this happens in a number of different networks in the body. There are different types of gap junction networks. We're talking about here chemical networks, not electrical networks, where we have formal synapses and we're establishing these pre and post synaptic relationships. So people have explored this in the past using different types of, just using data mining. And indeed, this uses some secondary data sets on synapses in the C. elegans connectome. The C. elegans connectome is good because we have the timing of the sort of the birth of these different neurons. And we have the synaptic data that talks about, that gives us information about which cells connect to what other cells and so forth. We have this both for the adult and in development. So in development is, of course, key because there are a lot of changes in, so we have the data across larval development. And so we can use really early larval connectivity as a proxy for what's going on when these networks first form. So in C. elegans, we have these connectomes that form, things get wired up. You get behaviors like muscle twitching that are autonomous from the connectome, and then later you get simple connectome related behaviors that start to emerge around the time that the egg hatches and going into this L1 phase of the larval period. And then during the larval period, you get a lot of synaptic rewiring before you get to the adult. So, it stands to reason that these synaptic connections not only, are important, but really characterize this first movement aspect, this first mover advantage. Because the first mover advantage will be very important for establishing, these connectome networks. Because as we know, connectome networks aren't sort of randomly connected. They're hierarchical. We have what we call rich club neurons. We have other types of peripheral neurons. And so that structure maybe is something we can describe with this. So this figure here is figure 2 from the paper. This is a diagram showing the different strategies for. So what happened in the paper was, you had some data for cellular development, for synaptic development, and those were brought together to determine these strategies and how this first mover advantage might play out. So these are observed strategies. So we have 5 strategies here. We have NP coupling, XNOR, XOR first mover, XOR second mover, and PN coupling. So So NP coupling is negative positive coupling. XNOR represents this sort of exclusive NOR logical rule. XOR first mover is exclusive OR for the first mover. XOR is exclusive OR for the second mover. And then PN coupling is where we have positive negative coupling. So what I mean by negative and positive is that we have these different, so each of these cells emerges at a specific time in development. So, in this little example, you might have like a cell that is born at 340 minutes of development, another cell that's born at 320 minutes of development, another cell that's born at, I think it's 350 minutes of development, and so on. And so they establish connections based on, say, the first cell to be born in that network sort of establishes the coupling. It establishes the axons that are sent out. It establishes the synaptic connections with later cells that emerge. And so we see for NP coupling, we see 320 and 350 are shaded. And for PN coupling, 330 and 360 are shaded. Or I think it's 360, 340, and 330. So this is, kind of, we can use these different rules to predict which cells will connect to what other cells. And so that first mover advantage is based on the age of the cell. When does it emerge in development? When does it differentiate? And so, we have this information. That's for NP and PN, so negative, positive, positive, negative. Then you have these logical rules applied to this sort of network where you have to connect to maybe something that emerges first or something that, emerges second. And you can apply, so exclusive or first mover, you have to have a connection. The first mover has to connect to the other, to its connecting cell. In exclusive or second mover, the later cell has to connect to the earlier cell. So there are actually rules and constraints on this. And these are tested in this paper. So this is the description here. NP coupling, this strategy is employed for roughly 40 to 50% of connections. NP coupling includes three other strategies, which are more focused aspects of the synaptic pairwise relationship. Exclusive or first mover is employed for around 7 to 8% of connections and can include both pre and postsynaptic partners born first. This exclusive or first mover strategy is a refinement of NP coupling, and then it excludes presynaptic partners born after the cell in question. Then we have exclusive or second mover, which is where it's a mixed strategy, so it's not a pure strategy. And what I mean by mixed strategies, it can include both pre and postsynaptic partners, so it has fewer constraints on it. It's employed for around 20% of connections, and exclusive or second mover only excludes postsynaptic partners born first. So there are different exclusions that are made to the data in kind of formulating these strategies and looking at how they explain the data. Then there's exclusive NOR, which is XNOR. In our sample, the strategy is exhibited by around 20% of connections. As an even more exclusive version of the exclusive OR strategies, XNOR strategies exclude only those connections where the presynaptic cell is born first and the postsynaptic cell is born second. And then finally, PN coupling, which is sort of the inverse of NP coupling, which so this category contains cells that have the opposite relationship between pre and postsynaptic cells relative to NP coupling. For cells deploying this strategy, on average, the presynaptic cells are born later than the postsynaptic partner. PN coupling is a pure strategy which does not overlap with any other strategies. We have a payoff matrix here which shows the different sorts of payoffs that you get for these different sort of first mover, subsequent mover, and random strategies.